Welcome to the Explainer. So, if you're tackling O level biology or honestly just kind of fascinated by the invisible machinery that's literally keeping you alive right now, well, you're in exactly the right place. Today, we're taking a microscopic journey. We're going to break down exactly how substances move across cell membranes and really visualize the mechanics behind diffusion, osmosis, and water potential. Okay, look at the big picture for a second. Think about taking a deep breath in. But now let's zoom way in, right down to the micro level, into your bloodstream. You see those exact oxygen particles rushing in and the carbon dioxide heading out? This effortless, totally invisible movement is happening millions of times a second inside of you. And it's all thanks to a process called diffusion. But, what are the actual mechanical rules driving this? Right, let's dive into our roadmap for today. First up, we've got the hidden world of diffusion. Second, concentration gradients. Third, osmosis and water potential. Fourth, we'll look at the osmotic effects on cells. And finally, number 5, we'll cover common mistakes and exam tips to really lock in that A. Let's kick things off with section 1, the hidden world of diffusion, the basics of particle movement. Here is the fundamental definition you absolutely need to lock in. Diffusion is the net movement of particles from an area of high concentration to an area of low concentration. See those densely packed particles on one side? Now follow those glowing vector arrows. That right there is your net directional flow. It shows the particles passively spreading out into that empty space, the area of low concentration. Think of it like being in a super crowded room, right? When a door opens to an empty room next door, people are naturally going to spread out just to get some personal space until both rooms are pretty much equal. Well, that's exactly what happens down in the micro world. Molecules move around completely randomly until they're evenly distributed. But here's a massive detail a lot of people miss. Even when they're perfectly balanced, they don't just freeze. No way. They keep moving, bouncing off each other. It's just that there's no longer a net movement in one specific direction. Moving right along to section 2. Driving force, concentration gradients. So think of the concentration gradient like a physical slope that particles just naturally want to roll down. And the rule of thumb? The steeper the slope, the faster the diffusion. As that slope gets steeper on screen, which represents a much more extreme difference in concentration, notice how those directional arrows actually thicken and get brighter. A steeper gradient physically cranks up the speed of diffusion. If that slope is completely flat, you've got zero net movement. All right, section 3, osmosis and water potential, the special case of water. Now, osmosis is basically a special type of diffusion, but it comes with stricter rules. It's strictly the movement of water from a region of higher water potential to lower water potential across a partially permeable membrane. Think of that membrane literally like a kitchen sieve. Those tiny blue water molecules, they effortlessly slip right through the gaps. But the larger, chunkier solute molecules get physically blocked. It's a totally selective barrier. But what actually is water potential? Well, it's just a measurement of the free energy of water molecules. Like, how free are they to just move around? Pure water has the absolute highest possible water potential. And in biology, we cap that maximum limit at exactly 0. See that big 0? Because there aren't any solutes getting in the way, 100% of those water molecules have the maximum free energy to move. But check this out. As soon as we start adding solutes, like salt or sugar, they interact with the water, and that actually reduces its free energy. This literally drags the water potential down from 0 straight into the negative numbers. Add a ton of solutes, and it plunges even deeper into the negatives. And because water always moves from higher to lower water potential, it's always going to rush away from that 0, straight down the slope and into those deep negative numbers. Which brings us to section 4, osmotic effects on cells, biology and action. Let's see this in action. Take an animal cell, like a red blood cell, and drop it into a dilute solution with high water potential. That's step 1. Now, step 2, because the inside of the cell has a more negative water potential, water just rushes in via osmosis. Step 3, the cell physically swells up, kind of like a water balloon filling too fast. And finally, step 4, because animal cells don't have any rigid outer structure to protect them, that immense internal pressure causes the membrane to just completely rupture and burst. Game over for the cell. But watch what happens when we swap that out for a plant cell. Step 1, it goes into that exact same dilute solution. Step 2, water rushes in. But wait, notice that thick structural boundary on the outside? That's the cell wall.
This rigid wall acts almost like a cage, structurally stopping the membrane from expanding too far. So instead of exploding, the cell just becomes incredibly firm, or turgid. In fact, this internal pressure is exactly why plants can stand upright and don't just wilt over.
So here's your ultimate study cheat sheet. An animal cell will burst in a dilute solution, but if you drop it in a highly concentrated sugary solution, water leaves and it totally shrivels up. Now, a plant cell, it's kept safely turgid by its cell wall in a dilute solution, but in a concentrated solution, it loses water too, and its membrane actually pulls away from the wall in a process called plasmolysis. It's a really beautiful contrast of how plant and animal biology handle osmotic stress.
And finally, Section 5, Common Mistakes and Exam Tips on How to Get an A. Okay, let's do a super quick, rapid-fire recap and debunk some common myths. First, remember diffusion is completely passive. Second, osmosis is just water moving to a lower water potential. Now, big mistake number 1, thinking particles just stop moving when they hit equilibrium. No way. Even when they're perfectly balanced, they are still bouncing around randomly. There's just no net movement. And mistake number 2, thinking diffusion requires active cellular energy. It absolutely doesn't. It is powered entirely by natural kinetic energy. Now for a massive exam tip. Practice drawing these diagrams explicitly. Don't just sketch a basic cell and call it a day. You've got to draw the exact directional vector arrows showing whether water is moving in or out. Clearly label that partially permeable membrane and mark it areas of high and low water potential. The examiner doesn't just want vocabulary. They want to see that you visually understand the dynamic flow. Think about this for a second. Every breath you take, the way your blood cells function, the way a massive oak tree stands perfectly tall, it is all driven by the completely invisible, purely random, passive motion of particles just seeking balance. It's essentially blind math and geometry, keeping biological life functioning. Kind of blows your mind, right? It really makes you wonder, if something this simple and microscopic is doing so much heavy lifting, what other invisible forces are secretly working behind the scenes, keeping you alive right at this very moment? Thanks for hanging out with me on The Explainer today. Keep questioning the invisible world, and I'll see you next time.